Okay, so here we have what's known as the synaptic cleft, and on either side we have the presynaptic neuron on the right and the postsynaptic neuron on the left. The action potential goes from the presynaptic neuron to the postsynaptic neuron, but how will it cross the synapse? As the action potential arrives at the terminus, these blue blobs that you see are protein channels, calcium ion channels, which are closed at the moment. But when depolarization occurs as the action potential arrives, this triggers these calcium ion channels to open, and thus calcium ions flood in. These white blobs that you can see here represent the calcium ions that have entered the axon via these blue protein channels. This triggers the vesicles containing the neurotransmitter acetylcholine to begin to move towards the membrane of the presynaptic neuron. Then, these vesicles fuse with the membrane of the presynaptic neuron. This allows the contents of the vesicles, the neurotransmitter acetylcholine, to diffuse across the synaptic cleft. On the membrane of the postsynaptic neuron, the sodium ion channels are closed. But now that the acetylcholine has diffused and has bound to them, they can open, allowing sodium ions to flood in. This causes depolarization of the new neuron, and so a new action potential is generated. The enzyme acetylcholine esterase breaks down acetylcholine into acetyl and choline. I'll explain that next. So, here you can see the acetylcholine esterase attached to the acetylcholine, breaking it up into two parts, acetyl and choline. Sodium channels closed, preventing any further depolarization. Acetylcholine can then be resynthesized and repackaged into vesicles ready for the next action potential to arrive. This uses ATP, which is supplied by mitochondria.